Assalamualaikum warahmatullahi wabarakatuh. My name is Aina Shafqat Azmi from S253, and today I will present to you about the synthesis of betulinic acid and hydrazine. My supervisor is Dr. Muhammad Tajuddin Ma'ali. So first we go to background of study. Betulinic acid is a natural substance from which tree that has many health benefits. Researchers are investigating its potential to treat cancer and its because it can kill tumor cells. It's also used in traditional medicine for stomach and intestinal issues. Researchers also are studying related compounds to improve its effectiveness and understand how it works. So this is the synthesis outline of my project, which is first there are two-step reaction which to synthesize betulinic acid hydrazine. The first reaction involves the esterification of betulinic acid and with diazomethane, and second is the amination using hydrazine. The problem statement here is betulinic acid has pharmacological and biological activity. It occurs naturally in plants but in limited quantities. Researchers study synthetic betulinic acid for health benefits. Betulinic acid and its derivative shows promise against cancer cells. Hydrazine derivative enhance its effectiveness. Synthesizing betulinic acid hydrazine through chemical reaction creates new compound with therapeutic potential. The research question here is the betulinic acid hydrazine can be synthesized using ester, and the second one is the solubility of betulinic acid hydrazine in H2O can be measured. The objective of this project is to synthesize betulinic acid hydrazine and to analyze all the synthesized compounds using spectroscopy analysis such as NMR, IR, and mass spectrometry. The significance of this study is researchers are trying to find the best treatment for cancer. They believe the betulinic acid could be a promising option because it has shown a strong anti-cancer effects in lab and animal studies. In this experiment, we are working to create betulinic acid hydrazine, which could be a powerful anti-cancer agent. We use different tests to make sure that the experiment successfully produce the compound. The expected outcome from this study is to produce the betulinic acid hydrazine, which contains its own benefits toward the biological activity. Apart from that, to produce a new product of betulinic acid derivative, which reaction occurs on carbon-28. Okay, so next we proceed to literature review. I have chosen 5 related journals that related to my project and to present here. First, we go from Law 2021. They say that if using soft lead extraction, it can yield concentration of betulinic acid ranging from 0.002% until 2%. While from Pinilla 2014, they said that when using supercritical fluid extraction, it can yield about 4.34% compared to using SLE, UAE, and PLE. Next, from Low also, they said that chemical synthesis of betulinic acid is essential and not relying only on natural extraction. And from UX 2012, they said that one of the advantages of betulinic acid is inhibit melanoma and consume potential anti-cancer effect. And the third one, this is one of the example of the chemical synthesis acid. Chemical synthesis betulinic acid is based from Kim from 1996. They said that betulinic acid can be synthesized from betulin that involves two-step process. First, it will undergo oxidation using Jones reagent, then undergo again reduction using sodium borohydride in THF. Lastly, betulinic acid can be combined with new compound to increase its biological activities. Based on Zhang 2015, they said that betulinic acid can produce twin drugs which combining with betulin to form dimers with anti-tumor properties, and acetylene compound 1 using acetic anhydride and reflux yields the anhydride of betulinic acid as a side product. Next, we proceed on the methodology of this experiment. In this method, it involves 5 steps of preparation, which is preparation of methylamine urea, preparation of nitrosyl methyl urea, preparation of diazomethane, preparation of betulinic acid ester, and preparation of betulinic acid hydrazine. First, we go to the preparation methylamine urea. In this step, the compound used in this method was methylamine hydrochloric acid and urea. After finished the synthesis, the compound it used for further reaction, which is preparation of nitrosyl methyl urea. Next, for the preparation of nitrosyl methyl urea, it involves 5 steps, which includes third and fifth step. If you can see from the slide, the final product in this preparation was yellowish in color. Next, for the preparation of diazomethane, it involves 4 steps to complete the full reaction. This method was done carefully as the products are easily to evaporate. We separate the compound using the separating funnel. We collected the upper product in yellow in color. Next, for the extraction of betulinic acid was not done by ourselves as it already have in our laboratory. Then we continue to prepare the betulinic acid ester using diazomethane that prepared before this. After complete the full reaction, we then send the sample to analyze using 1H NMR and 13 carbon NMR. Next, lastly, the final step for this project was preparing the betulinic acid hydrazine. It took a shorter time to complete the full reaction compared to preparing betulinic acid ester. After complete the full reaction, we send the sample to analyze using 1H NMR, 13 carbon NMR, ATR, FTIR, and also GCL test. Then we proceed to discuss on the results. Here is the result for my project. For betulinic acid ester, and betulinic acid hydrazine, we use 1 NMR and 30 carbon NMR to analyze the sample. But there is extra analysis for betulinic acid hydrazine, where we use ATR, FTIR, and also DCMS. So here is is the result of 1 NMR betulinic acid. 1 NMR is providing information about the hydrogen atom in a molecule, revealing their specific environments and chemical quality. While on the other hand, 13 carbon NMR more focused on the carbon atoms, showing the unique chemical environments. So here is the 1 NMR of betulinic acid. In the 1 NMR, we can see the peak represent the unique hydrogen environments in its pentacyclic triterpenoid structure. The spectrum displays signals for hydrogen atom in various functional groups such as hydroxyl OH, methylene CH2, and methyl CH3 groups. Each peak corresponds to the specific arrangement of hydrogen atoms in the betulinic acid molecule. You can see these are the peaks that appear on the result. Next, for the 13 carbon NMR spectrum of betulinic acid, the peak represents the unique carbon environment in its pentacyclic triterpenoid structure. The spectrum displays signals for carbon atoms in various functional groups such as carbonyl C-double-bond-O, olefinic C-double-bond-C, and aliphatic CH2 and CH3 carbons. Each peak corresponds to the specific arrangement of carbon atoms in the betulinic acid molecule. There are 3 carbons that we focus more on betulinic acid, which carbon 20, carbon 3, and carbon 29. In the carbon 20, it is a part of pentacyclic triterpenoid skeleton of betulinic acid. The chemical shift and peak intensity of carbon 20 provide insights into the connectivity and arrangement of the rings in the triterpenoid structure. While for the carbon 3 is another crucial carbon atom in betulinic acid. This carbon is part of the carboxylic acid group C-double-bond-O in the molecule. The chemical shift of the carbon 3 is indicative of the electronic environment and the chemical bonding around the carboxylic acid functional group. Carbon 29 is involved in the carbon 28 methyl group of betulinic acid. The chemical shift of carbon 29 provides information about the electronic environment and spatial arrangement of the methyl group, contributing to the overall molecular structure. Next, we move to betulinic acid ester. On 1H NMR, spectrum of betulinic acid ester will show additional peaks compared to betulinic acid due to the presence of an ester functional group. The esterification modifies the specific proton environments in the molecule, resulting in new signals associated with the ester group. The characteristic of peak of betulinic acid's triterpenoid structure is still present, but the ester signal is distinguishable. At the peak 3.5 until 3.9, if you can see here, there is a new peak that indicates there is presence of ester group. Then for the 13 carbon NMR, spectrum of betulinic acid ester will also have additional peaks compared to betulinic acid ester due to presence of ester functional group. The esterification introduced new carbon environments, resulting in new specific to the ester group. The characteristics peak of betulinic acid triterpenoid structure also present, but the ester signal is distinguishable. As you can see at carbon 28, the peak appear more upfield compared to the peak at carbon 28 of betulinic acid. Here is the presence of the ester group. Next, we go to the betulinic acid hydrazine, the result, which on 1H NMR. 1H NMR spectrum of betulinic acid hydrazine will also have a unique peak introduced by the hydrazine moiety. The addition of hydrazine functional group NH and H2 alters the proton environment in the molecule, leading to a new signal specific to the hydrazine moiety. The spectral data display the characteristic peaks of betulinic acid as well as additional signals associated with hydrazine group. Next, for the 30 carbon NMR, spectrum of betulinic acid hydrazine will also display the unique peaks introduced by the hydrazine moiety. The addition of hydrazine functional group NH and H2 creates a new carbon environment specific to the hydrazine group. The spectral data will show the characteristics peaks of betulinic acid along with the additional signals associated with the hydrazine moiety. As you can see, at carbon 28, the peak appears more upfield compared to the peak at carbon 28 of betulinic acid and betulinic acid ester. Next, we go to the IR spectrum of the betulinic acid hydrazine. IR infrared radiation is used to probe the vibration of molecular bonds, allowing the identification of specific functional groups present in the compound. The presence of a strong absorption band around 3367 cm-2 in the IR spectrum indicates the presence of NH stretching, which is the characteristic of the hydrazine moiety. This peak confirms that the hydrazine group has been successfully incorporated into the betulinic acid molecule. Additionally, a sharp peak at 1703 cm-1 suggests the presence of a carbonyl group C-double-bond-O, which is also a characteristic feature of betulinic acid. The disappearance of carboxylic acid peak around 700 cm-1 further confirmed that the formation of the hydrazine derivative as the carboxylic acid group is involved in the reaction with hydrazine using the synthesis. Lastly, the result for betulinic acid hydrazine, which is the GCMS mass spectrometry, Mass spectrometry is a powerful analytical technique used to determine the molecular weight of a compound and provide the information about its chemical structure and composition. In the context of betulinic acid hydrazine synthesis, mass spectrometry helps in confirming the successful formation of the desired product and provide the insight into its molecular characteristics. In mass spectrometry, the molecular weight of the synthesized compound can be determined. The mass spectrometer ionizes the molecules and separate them based on their mass to charge ratio. The molecular ion peak M+ in the mass spectrum represents the molecular weight of the compound. By analyzing the mass spectrum, researchers can confirm that the molecular weight of the synthesis betulinic acid hydrazine match the expected value based on the reaction stoichiometry, providing further evidence for the successful synthesis. The original molecular ion was supposedly to be 470.4, but the result we get was 488, which indicate the molecular ion of betulinic acid hydrazine. There is an addition of ammonium group. Lastly, for the conclusion and recommendation for this project. In conclusion, we have successfully synthesized betulinic acid ester and betulinic acid hydrazine. This synthesis holds promise for creating a new compound with potential therapeutic benefits. Analyzing the NMR spectra confirms the presence of the specific functional groups, including the hydrazine moiety. And it also helps to explore the medicinal application. The hydrazine derivative of betulinic acid shows the improved solubility and stability, making it more valuable candidate for the drug development. For the recommendation, further research and studies are needed to fully explore the potential of betulinic acid and hydrazine derivative, evaluate their pharmacological activities, identify the promising compound for specific therapeutic uses. Efficient and scalable synthetic methods should be developed to make the production of betulinic acid hydrazine derivative more economically viable, and exploring alternative reaction, condition, and eco-friendly solvent can enhance overall efficiency and yield. Exploring further reaction and chemical modification of betulinic acid hydrazine also can enhance its pharmacological properties and therapeutic potential by selectively modifying the functional group at a specific position to optimize its biology and interaction with the target biomolecules, ultimately improving the efficacy against various diseases. Then we go to the references. These are my references that I use in this presentation. And that's all for me. Thank you for lending your ears and have a good day.